Hello and welcome. My name is Victor Muya and this is Project 2201 of this year's Summer of HPC. The title of the project is Leveraging HPC to Test the Efficiency and Scalability of a Genetic Analysis Tool. Organisms of different species interbreed. Their offspring are said to be hybrid and are heterozygous since they carry copies of genetic material from both parents at each site in their genome. This creates an imbalance in the hybrid's DNA since the two copies of DNA are not fully identical and cannot pair easily during cell division. As a result, some of the alleles in the hybrid's heterozygous sites are lost or deleted in what is known as loss of heterozygosity, or LOH as I will refer to it in this video. At the moment, little to no software exists that can test genomic data for LOH, and to fill this gap, the Comparative Genomics Lab at the Barcelona Supercomputing Center has developed JLO, a genetic analysis toolkit that extracts blocks of LOH from sequencing data. Starting with single nucleotide polymorphism data, JLO identifies regions in a genome where stretches of the difference between two genomes have been lost during evolution through LOH. JLO's core is written in Python, but it also comes with a pipeline written in Nextflow, which enables it to scale on a cluster. So to analyze our genomes, first we establish the genome sequences through DNA sequencing, and this data is passed to a computer in the form of text files with the letters A, T, C, and G representing the individual bases. In hybrid mode, the JLO algorithm needs three inputs: two FASTA files storing the reference sequences of the parent species of the hybrid, two BAM files mapping the records of the hybrid reads onto the parental genome, and two VCF files representing the variance between the sequencing reads of the hybrid and the two parental genomes. In default mode, JLO works with a single file of each type. The sample we're using for testing is a hybrid of the yeast species Saccharomyces cerevisiae and Saccharomyces ovarum. The raw sequencing data comes in FASTQ format and it can be passed to the next flow pipeline and we would still get the desired output. With these input files, JLO outputs blocks of the two parental genomes where some alleles have been lost using a series of several files, which together with the BAM files can be viewed in a genomics viewer and would look something like the visualization at the bottom of the slide. Now to the testing and optimization, which is where I come in. JLO has a few modules to extract and manage LOH blocks, but I mostly worked on testing JLO's extract module as it is a primary module in the toolkit. When run in default mode with one FASTA file, one VCF, and one BAM file, this is what gets printed on the terminal. And each of these stages is the functions that JLO goes through when running the analysis and their results, each of which are printed at each stage on the terminal, as you can see here, and in the eventual output files. I was running the tests as job scripts on the Mianostrom supercomputing cluster, which was able to compute the analysis very quickly. Using a cluster also helped me create different scenarios when running the program. For example, different number of CPU cores and different amounts of RAM. And then I observed how the program behaved under these different conditions. First step in testing the efficiency of JLO was profiling, where using different Python tools, I measured the amount of time and memory each of the individual functions within JLO extract took to run. With these, we were able to identify the functions that consume the most runtime and try to optimize them where it was possible. Here's a summary of the time profile for JLO extract visualized in SnakeViews, showing the amount of time consumed by each of the functions in the extract module. When testing whether JLO is scalable based on computational time, I ran the program with subsets of different sizes with a fixed number of CPU cores and memory, each time measuring how much time it took. I used the subsets from our test samples, split them into subsets of incremental size and ran them as job scripts on the cluster. And the results are as shown in the graphs below. On the left is a bar graph showing the size of the datasets in megabytes for one chromosome, 4 chromosomes, all the way up to the entire set, which was 17 chromosomes. And the second graph shows how much time it took to extract the LOH block from these sets. In the second graph, there are two lines. One represents real time, which is red, and the blue one represents user time. Real time is the actual time from when we run the command up until we get the output. And user time is the cumulative time taken by the CPUs to run the program. You will notice that user time is relatively higher than real time because JLo leverages the power of multiple CPUs by using multiprocessing extensively in its functions to run its analysis. The computational time increases linearly, as you can see the line graph, as we increase the size of the datasets, showing that JLo is scalable by computational time. Tested using different numbers of CPU cores and 12 chromosomes of the sample genome data, the real time decreases as we increase the CPU cores and flattens out when we reach about 4 CPUs for this dataset, which shows that for this set, 4 CPUs are enough to run the program and give us our output. Overall, JLo performed well across the different tests that I conducted and proved to be scalable with increasing data sizes and CPU cores and does not consume a lot of memory to give the required output. In conclusion, the summer of HPC has been an enriching experience for me. I have enjoyed working on this project and learned a lot about genomics, code profiling and testing, and how to work on an HPC cluster. I would encourage anyone with an interest in computing to apply and participate. Thank you.